Hello, this is Lesson 7, the role of enzymes. DNA replication is carried out by a series of enzymes. The first unzip the molecule of DNA by breaking these weak hydrogen bonds between the base pairs, unwinding the two strands of the molecule. Each strand serves as the template for the attachment of complementary bases. The principal enzyme involved, and you need to know this, in replication is called DNA polymerase. DNA polymerase is an enzyme that joins individual nucleotides to produce a new strand of DNA. This DNA polymerase is also cool because it proofreads each DNA strand, ensuring that each molecule is a perfect copy of the original. Telomeres. The tip of the chromosomes is known as telomeres. The ends of DNA molecules located at the telomeres is particularly difficult to copy. Over time, DNA may actually be lost from the telomeres each time the chromosome is replicated. An enzyme called telomerase compensates for this problem by adding a short repeated DNA sequence to the telomerase, lengthening the chromosomes slightly by making it less likely to that an important gene sequence will be lost during replication. And that's kind of how it looks. The very end of the chromosome, or is the telomeres, the ends. How does DNA replication differ in prokaryotic cells and eukaryotic cells? Replication in most prokaryotic cells starts from one single point and proceeds in two directions until the entire chromosome is copied. In eukaryotic cells, it can start at dozens or hundreds of different places on the DNA molecule and proceeding in different directions until it's copied. The cells of most prokaryotic cells have a singular cellular DNA molecule in the cytoplasm containing nearly all of the genetic information. Eukaryotic cells, on the other hand, have up to 1,000 times more DNA, so you need something more efficient that's going to help copy the DNA. In most prokaryotic cells, DNA replication does not start until the regulatory proteins bind to a single starting point of the chromosome. This triggers the beginning of DNA replication. Replication in most prokaryotic cells starts from a single point and proceeds in two directions until the entire chromosome has been copied. So one point and in two directions. Often, two chromosomes produced by the replication are attached to different points inside the cell membrane and are separated when the cell splits into two new cells. In eukaryotic cells, this replication can start at dozens or even hundreds of different places in the DNA molecule, proceeding in both directions until the chromosome is completely copied. Two copies of DNA produced by replication remain closely associated until the cell enters prophase of mitosis. At that point, the chromosomes condense and two chromatids in each chromosome become clearly visible. They separate from each other in anaphase and mitosis, producing two cells, each with a complete set of genes coded in DNA. Thank you for listening.